Sarah and Emily were twins, identical in every way, or so everyone thought. Sure, they looked the same: fiery red hair, bright green eyes, and a dusting of freckles across their noses. But their personalities, night and day. Sarah was a whirlwind of energy, always up for an adventure, scaling trees and building forts in the backyard. Emily, on the other hand, preferred the quiet comfort of a book, her nose buried in stories of fantastical lands. Their parents scratched their heads. They raised them the same way, provided the same experiences, yet somehow they ended up so different. This sparked a question in Sarah's mind. How could they be so alike yet so different? The answer, she later discovered, lay in a fascinating field called epigenetics. Epigenetics, she learned, wasn't about changing the DNA code itself, but rather how that code was read and used. Imagine a library with millions of books, the genes, and epigenetics as the system that determines which books are readily available and which are tucked away on dusty shelves. It turns out the environment plays a crucial role in this system. The food you eat, the stress you experience, even the things your parents were exposed to before you were born. All of these can leave tiny marks on your genes, influencing how they are expressed. So, could the reason for Sarah and Emily's differences be the subtle variations in their environment? Perhaps Sarah's adventurous spirit stemmed from an early exposure to the outdoors, while Emily's love for books was nurtured by quiet evenings spent curled up with their mother. It was a fascinating possibility. As Sarah delved deeper, she discovered the vast potential of epigenetics. It wasn't just about explaining why twins were different. It was about understanding how our environment shapes our health and well-being. From the impact of diet and exercise on gene expression to the influence of early life experiences on mental health, epigenetics offered a whole new lens to view human biology. This knowledge ignited a passion in Sarah. Maybe, just maybe, by understanding epigenetics, we could unlock ways to promote healthy gene expression, potentially mitigating the effects of disease and even influencing future generations. Just like Sarah and Emily, with identical DNA, could develop different traits due to subtle variations in their environment, epigenetics revealed a whole new way to understand human health and disease. Epigenetic delves into how environmental exposures can engrave subatomic signatures on genes, possibly reverberating across generations. This worldview challenges traditional ideas of genetic inheritance, highlighting the powerful role of environmental influences in shaping gene expression. In this video, we will discuss the key things you should know about epigenetics, helping you understand what it is about and possibly have a better idea of who you are and what you can be. Epigenetics versus traditional genetics. Traditionally, genes were thought to be fixed and determine our traits. Epigenetics challenges this by showing how the environment can influence gene expression without changing the DNA sequence itself. Let's picture Sarah and Emily as seeds. Their DNA, the blueprint for their entire being, is like the complex instructions encoded within each seed. These instructions hold the potential for a sunflower or a daisy, depending on the specific DNA sequence. Traditionally, genetics was like believing that these seeds were predetermined. One seed destined to be a sunflower, the other a daisy. Epigenetics, however, throws a twist into the story. It's like planting those seeds in different environments. Sarah, the adventurous twin, might have been planted near a lively stream, bathed in sunlight, and constantly exposed to wind. Emily, the bookish twin, might have settled under a shady tree, sheltered from the elements and surrounded by the quiet rustle of leaves. Despite having the same genetic instructions, the same seed, environmental factors such as sunlight, wind, shade plays a crucial role in how those instructions are read and used. The sunflower seed, exposed to the open environment, might grow tall and strong, its genes for height and sun absorption being expressed more actively. The daisy seed, under the shade, might prioritize spreading its roots and remain smaller, with genes for root growth taking precedence. Just like the environment shapes how the sunflower and daisy seeds grow, epigenetics reveals that our environment can influence how our genes are expressed. The DNA code remains the same, like the seed instructions, but the way those instructions are interpreted and used can be influenced by what surrounds us. So Sarah and Emily, with identical DNA, could develop different traits due to the subtle variations in their environment, like sunlight and wind for Sarah and shade and quiet for Emily. This is the essence of epigenetics. It's not about changing the seed itself, but rather the way it grows. Types of epigenetic modifications. There are three main mechanisms: DNA methylation, histone modifications, and non-coding RNAs, and each plays a role in regulating gene activity by affecting how tightly DNA is packaged. Let's go back to the story. Imagine Sarah and Emily's genes as a vast library filled with millions of books, holding all the information needed to build and run their bodies. Epigenetics, in this analogy, isn't about rewriting the books themselves, the DNA code, but rather about the different ways those books can be accessed and used. If you don't understand, let me explain how the three main epigenetic mechanisms play a role. 1. DNA methylation. Picture a librarian meticulously adding tiny stickers to specific books on the shelves. These stickers we will call methyl groups. These stickers don't change the content of the books, but they might make them harder to open and read. In DNA methylation, adding methyl groups to specific genes can make them less active, essentially putting them on a dusty shelf in the library. This might explain why Emily, perhaps exposed to a quieter environment, has some genes related to social interaction methylated and less active, leading to her preference for solitude. 2. Histone modifications. Still using the library analogy, the books are wrapped around spools called histones. These spools can be decorated with different chemical tags, aka modifications. Some tags might loosen the way the books are wrapped, making them easier to access and read, while others might tighten the wrapping, making them harder to reach. In histone modifications, adding specific tags to histones can loosen the DNA around them, making the genes more accessible for protein machinery to read and use. This could be happening in Sarah, where exposure to a stimulating environment might have led to modifications on genes related to physical activity, making them more readily expressed and contributing to her adventurous spirit. 3. Non-coding RNAs. Think of these as special hint books in the library that don't contain information themselves but guide the librarian, the protein machinery, towards specific sections, genes, in the main books, DNA. Non-coding RNAs can interact with DNA and proteins, influencing how genes are accessed and used. While the exact mechanisms are still being unraveled, some non-coding RNAs might flag specific genes for activation, while others might mark them for silencing. These non-coding RNAs could be another player in shaping Sarah and Emily's personalities. Perhaps certain non-coding RNAs in Sarah's cells are promoting the expression of genes related to movement and exploration, while in Emily, different non-coding RNAs might be prioritizing genes associated with focus and concentration. Understanding these different epigenetic mechanisms is like having a better understanding of how the library is organized and how the librarian retrieves information. Epigenetics reveals the fascinating ways our environment can influence how our genes are accessed and used, ultimately shaping who we are, even if we share the same basic genetic code like Sarah and Emily. Environmental influences on epigenetics. There are various environmental factors that can shape epigenetic patterns. These include pollutants, diet, exercise, stress, and even early life experiences. Again, let us picture Sarah and Emily's environment as a constant stream of colorful paint splatters. These splatters represent the various environmental influences that can leave their mark on how genes are expressed. Let's break down how these various environmental influences might have played a role in shaping their personalities. Pollutants. Tiny paint splatters of pollution from car exhaust or industrial fumes might have landed on some of Sarah's genes. These could potentially influence genes related to lung function or immune response, subtly shaping her health in the long run. Diet. Sarah's adventurous spirit might have led her to explore different foods, represented by a vibrant splash of colors on her genes. This nutritional paint could have activated genes related to energy metabolism or even brain development, contributing to her active lifestyle. In contrast, Emily's preference for quiet evenings spent indoors might be reflected in a calmer paint splatter on her genes. Perhaps a focus on healthy whole foods, represented by greens and browns, has influenced genes associated with gut health or cognitive function, supporting her love for reading and learning. Exercise. Sarah's constant movement and outdoor adventures could be like a burst of energetic paint splatters on her genes. This exercise paint might have activated genes related to muscle growth or cardiovascular health, contributing to her athleticism. Emily's quieter pursuits might be reflected in a more subtle paint splatter on her genes. Perhaps a focus on activities like yoga or meditation, represented by calming blues and purples, has influenced genes related to stress management or focus, supporting her introspective nature. To summarize, the environment acts like a paintbrush, leaving colorful marks on our genes. These marks, even the subtle ones, can influence how our genes function, shaping who we become. The significance of epigenetics research. The potential of epigenetics to revolutionize our understanding of health and disease can't be overemphasized. It offers insights into intergenerational effects, the nature versus nurture debate, and the development of new therapies. The best way to look at it is to liken it to a vast library filled with countless books holding the secrets to human health and disease. Traditional genetics for a long time was like focusing solely on the DNA code written in each book. This only offered valuable information, but it didn't tell the whole story. Epigenetics research, however, is like discovering a whole new section of the library, one filled with colorful bookmarks and annotations. These bookmarks and annotations represent the epigenetic modifications that influence how the genes or you'd like the books are used. Epigenetics research sheds light on two long-standing questions and opens doors for future treatments. Firstly, it explains how the environment can influence health across generations. Imagine children inheriting not just their parents' genes, but also epigenetic modifications, like bookmarks marking important passages. These modifications can explain why certain health risks run in families even without changes in the DNA itself. Secondly, epigenetics bridges the nature versus nurture debate. Our genes, the books, hold the potential for various traits, but the environment, the bookmarks, ultimately decides which ones are expressed. This intricate interplay between nature and nurture offers exciting possibilities for new therapies. By understanding how environment influences gene expression, scientists might rewrite these bookmarks and develop treatments to prevent or manage diseases. Epigenetics research is like having a powerful new tool in the library of human health. It allows us to go beyond the DNA code and explore the fascinating world of how our environment shapes who we are. When we understand these epigenetic modifications, we can unlock new insights into the causes of diseases, potentially influence intergenerational health, and even pave the way for the development of revolutionary new therapies. The field of epigenetics holds immense potential for understanding how genes are passed down with environmental influences. Continued research is essential to navigate this complex area and ensure the well-being of future generations. Want to dive deeper into the exciting world of genetics and mental health? Subscribe to my channel and join our community. We'll be exploring the latest discoveries together. See you next time.